So welcome to Accessory Academy. So in this we are starting with the methods in biology, our unit 13 of CSI syllabus. So this also side by side हम करते रहेंगे. Okay. So first thing is about the purification of DNA and RNA. I'm sure you must have done in your MSc project, but we will be doing with detailed steps like Q. And I have put certain videos also for better understanding in case you have not done in the lab. Okay, so before I proceed, how many of you have done in the lab? Whether you have used the kit or the conventional method or not done. Yes. Have you guys done any time DNA purification?
No, ma'am, I haven't.
Okay. What about Akshay and Shruti?
We just did experiment on gross gel electrophoresis, like the restriction digestion part. We
Acha.
didn't do purification.
Acha. So DNA isolation is not done. Okay. It's okay. So like initially in BSc and all, if you would have done, maybe the conventional method was given. Ki plant mein se nikalo, because like plant you can get a huge amount of DNA, right?
DNA purification, we haven't. We have isolated DNA and RNA though, like the Orsinol method and everything.
No, I'm talking about purification. Isolation में क्या
Yeah,
होगा?
purification hasn't done. Purification नहीं हुआ है. Just isolation में.
So isolation means you will have the other contamination also along with that.
Yes.
So I was talking about the purification. Anyways, so I'm sure you must be aware of the steps which are being involved. Okay. Now, जो methods in biology है, जो भी हम करेंगे, the important part about about the methods in biology is that how and when to use which technique. ठीक है. जब भी इंटरव्यू होता है या क्वेश्चंस आता है, आजकल तो पार्टशीट में फिर भी मेंशन कर देते हैं दैट दिस इज फ्रॉम द सीडी का क्वेश्चन है या एनएमआर का क्वेश्चन है. ठीक है. देयर माइट बी चांसेस जहां पे मेंशन नहीं किया है. सो यू नीड टू अंडरस्टैंड अगर ये पर्टिकुलर क्वेश्चन पूछा है तो कौन सा इंस्ट्रूमेंट यूज होना चाहिए. ठीक है. सो having the knowledge of like which instrument to be used when and how is more important than knowing how to use the instrument. ठीक है. कोई भी इंस्ट्रूमेंट है, जैसे कि सपोज अगर मैंने कोई लैब खोली है एंड इफ आई एम लुकिंग लाइक ए रिसर्चर एक्सपीरियंस पर्सन, तो उसने कुछ और कंपनी का यूज किया होगा, उसको मेरे कंपनी का यूज नहीं आता होगा, राइट. तो जो भी बंदा जब भी लैब में ज्वाइन करता है वो हमेशा ट्रेनिंग पीरियड में जाता ही जाता है, भले उसके पास 10 साल का एक्सपीरियंस हो. ठीक है. इट जस्ट दैट इंस्ट्रूमेंट फ्रॉम कंपनी टू कंपनी इट वेरीज. आपको मालूम है एक नया मोबाइल सीखने में भी कम से कम एक दिन तो लगता है, इवन दो यू आर एक्सपर्ट इन यूजिंग द स्मार्टफोन्स. एक दिन तो लगता है कि हाँ उसके फीचर्स पता करने में. सेम वे व्हेन यू जॉइन एनी लैब, डोंट थिंक दैट कि मुझे कुछ नहीं आता है. दे आर गोइंग टू टीच यू. हर एक इंस्ट्रूमेंट का अपना अपना तरीका होता है, हर एक लैब का अपना कल्चर होता है, लाइक हाउ टू यूज एंड व्हेन टू यूज. So you will be trained for that, but the basic understanding ये होनी चाहिए when to use and what. ये आपको कोई नहीं सिखाएगा. ठीक है. So that is what we're going to learn in this session now. So before starting any genetic engineering project, it is always reasonable to have a pure amount of DNA. Okay. Later, which can be cut with the help of the restriction enzymes and ligated so that you can have a new gene, or you can go for the cloning. So there are many companies which makes the DNA extraction and purification kit. You must have heard also. We have got the Novozymes, Thermo Fisher hai, Abbott hai, BioEra, like many are there. अभी तो बहुत सारे आ गए हैं. So the basic procedure requires the releasing of the DNA from the cells and purification of the DNA so that you can use in the experiment. So there are main steps: cell lysis, enzymatic treatment, purification of DNA, and finally the quantification of DNA. So purification of DNA, we are going to discuss both the conventional method as well as the kit method. Okay. So today first we'll talk about the cell lysis. Even though we say that it is easy, but there are many things, many important points which needs to be understood in this case. So first let's talk about the cell lysis. So there are many methods are there. First, always we need a starting material. Okay. So your starting material could be anything. It can be a bacterial cell, yeast cell, or it can be an animal cell, plant cell. So depending on that, you are going to have the different methods of cell lysis. Okay. Now, Okay, so let me just finish this cell lysis and then I'm going to ask certain questions, which is like already asked in the exams, and maybe you can also ask me the questions. So first you need to have the starting material. Now, depending on the starting material, you can start doing the cell lysis, and always make sure that the cell solution is always, freshly harvested, or it is kept frozen. Now, why we need freshly harvested or frozen? Frozen bhi matlab it's not like 20 degrees Celsius or minus 20 degrees. It should be either minus 80 or minus, not minus, or it should be dipped into the liquid nitrogen. So liquid nitrogen is minus 196 degrees Celsius, right? So itna frozen hona chahiye, or at least it should be freshly harvested. Why it is? In order to avoid the degradation by the enzymes. Okay? Otherwise, what will happen? There are chances that the things in which you are interested is already being degraded. So आप जो भी काम करोगे सारा waste जाएगा. Okay. So we have got there are different types of cell destruction methods. So we have got mechanical and non-mechanical. So can you name like what are the different mechanical methods? कोई बता सकता है? So non-mechanical, we have got the chemical-based method and enzyme-based methods are there. What about the mechanical?
Mortar and pestle.
Yes, very good. Mortar and pestle is there, and ये हर लैब में यूज होता है. Then
Homogenizer.
yes, very good. Homogenizer. And
Centrifugation.
I didn't hear once again.
Centrifugation.
Centrifugation. Ha. So it has to be ultra then. Centrifugation. Then
Lyophilizer.
lyophilizer ऐसा होगा. See, lyophilizer, then again you need to go with the mortar pestle then. आप उसको पाउडर बना सकते हो lyophilizer में. You can remove the excess of water, then it becomes a powder, then again you come back to the mortar pestle only. You have got the French press. Okay. Then we have got the sonication also. Okay. So of these in the lab, all these are used depending on your source of the cell. Okay. So anyways, I will discuss, and ऐसा नहीं कि सिर्फ mechanical है तो सिर्फ mechanically use किया है. It is also coupled with the chemical and with the enzymatic. We'll come to that. Okay. So we have got the mechanical methods, so in which we are making use of the force in order to separate out the intracellular protein. So mortar pestle हो गया, blender हो गया, bead beating, yes, ultrasonication and homogenizer. So mortar pestle everyone has used, right? Now just imagine if suppose अगर मैं किसी bacterial cell पे काम कर रही हूँ या plant cell पे काम कर रही हूँ, so what is the first thing I will be doing? First thing is that you are going to take the weight of the sample. Okay. तो यहाँ पे आप plant के case में you can take the weight of the plant tissue, whereas in the case of the bacteria, what you can do is you have got the bacterial suspension. Okay, bacterial suspension रहेगा. At least you will have the absorbance at 600. कितना है उसका? That much you have decided. ठीक है, मैं 10 ml of bacterial suspension at a absorbance 600.8 पे मैंने वो सारे cells लिए हैं, saying that this is in the logarithmic phase or exponential phase of the bacteria. ठीक है, active phase of the bacteria. मैंने उसमें से total 10 ml लिया है, इसको मैंने centrifuge किया, and whatever I'm gonna get now put into the liquid nitrogen, and then liquid nitrogen के बाद मैंने mortar pestle में डाल के उसको जो भी किया, ठीक है, grind किया. Now what I'm gonna get is the required proteins and DNA. So depending on the requirement, I'm gonna add the buffer and go for the further steps. Is this much part clear? If it is bacteria, मैं कैसे steps आगे बढ़ूंगी? ये clear हुआ? Like how I'm gonna do?
Repeat it.
Oh, okay, I should repeat. Huh? Okay, I'll go slowly, maybe getting confused. Okay, so if I'm using mortar pestle method and my cell over here is bacterial cell. Okay, now always you need to have a reference, like starting position. So suppose it was plant. I'm going to take around 100 gram of plant tissue. and then dipped into liquid nitrogen so that it becomes hard. Then I'm going to put into this and grind it. Okay. Then add buffer and take into the centrifuge tube or whatever sample. Okay. Now I'm going to do the further steps of the protein or DNA purification. This much is clear. Like अगर plant का case है तो मैं कैसे करूँगी? Is this much is clear?
Yes, ma'am.
So this 100 gram is the reference. Maybe your lab is doing with 20 gram or with doing with 100 gram actually ज़्यादा ही हो गया. 20 gram हो गया या 50 gram हो गया, depending on the requirement. Okay. What I meant is that you should know from where you have started. ऐसा नहीं कि मैं पूरा का पूरा एक पत्ता या एक स्टेम लेके शुरू कर दूंगी, ऐसा नहीं होता है. You have to have some reference point कि हाँ, I have started with 100 gram of a sample and at the end I got around 50 nanogram of your DNA or I got a 1 milligram of protein. Okay, so I need a reference. Okay, I started with 100 gram and I got this much of DNA and this much of protein. This is in the case of plant. Okay, now let's talk about bacteria. So, so any bacteria, if I am interested to study the proteins of the DNA of bacteria, the first thing is absorbance at 600 nanometer. It should be from 0.8 to 1 OD. 1 के ऊपर हम लेते नहीं है because it is not going to come into the working range. So we don't consider above 1. So we are going to take around 0.08 to 1. This is the your logarithmic phase of the bacteria. ठीक है? यानी कि we say that the actively growing bacteria are present. Now, of course, in my conical flowers, whenever we grow, at least 100 ml हम कल्चर करेंगे. तो मैंने क्या किया है, उस 100 ml में से I have taken only 10 ml of bacteria suspension. This ml, okay, I'm going to centrifuge. So what will happen after the centrifuge? The bacteria pellet I'm going to get over here, correct? And the media is going to be removed. Is this much clear? हम 10,000 RPM में 10 minutes के लिए centrifuge करेंगे. So what will happen? At the end, I'm gonna get the pellet. ये pellet क्या है? मेरे cells से. जो ऊपर media है, मैंने remove कर दिया. Now, take the weight of this. Now, add liquid nitrogen to this. What will happen liquid nitrogen? इसको वो solid बना देगा, एकदम hard like rock. Put into the mortar pestle, grind it. Okay. So now what happens, since from liquid nitrogen to room temperature, it will start melting. It will become a liquid. Take that liquid. It is nothing but your protein and DNA suspension. Clear?
Yes, ma'am.
हाँ, अभी clear है? तो even though I just said mortar pestle, but उससे पहले कितने सारे steps हैं हमारे पास करने को. It's not like आपको किसी ने हाथ में cells दे दिए. You have to get all these steps are required. Okay. Okay. So this is one mechanical method of disrupting the cell. The others are blenders. Okay. So you can use high speed blenders in order to disrupt the cell walls. So again blenders हम किस में use करेंगे? In the case of the bacterial cells. यहाँ पे भी cell wall आ रहा है. ठीक है. Now, bead beating क्या होता है? Where you have got glass beads, okay? Glass beads, sorry. अब glass beads का कैसा है? In this glass beads, you are going to add your culture. तो वो यहाँ पे ऐसे से आएंगे. So because of the centrifugation and agitation, beads के बीच में आके cells are going to get lysed. Okay, so this is your bead beating. Okay, clear? What is bead beating? So ये कुछ नहीं, powder जैसा दिखेगा आपको. Just put into your cell suspension and put into the vials. Put it into the centrifuge. Now because of the agitation, it is moving so fast. Because of that agitation, the glass beads are going to rub to each other. और जैसे बीच में कोई cells आएगा, it will get lysed. At the end, just centrifuge, supernatant आप ले लोगे. Supernatant में आपका protein रहेगा, नीचे आपका cell debris रहेगा. Clear? This is your bead beating. Guys, stop me अगर नहीं समझ में आ रहा है. Then sonication. So sonication, you are making use of the sonic waves over here. So ultrasonic homogenizers, they work by inducing the vibration in the titanium probe, which is immersed in the cell solution. So what you do over here is you take a you take a beaker, आप उसमें ice डालते हो, ठीक है, and उसके अंदर आप अपना centrifuge tube डालते हो, इसमें आपका cell suspension है. Then probe insert करोगे, this probe is going to generate the ultrasonic waves, जिसके वजह से the cells are going to get disrupted. Now the question is, why are you keeping on ice? Ice की क्या जरूरत है?
This process may induce heat so that to maintain the organelles and whatever we want in the suspension intact, we need ice.
Yes, very good, very good. So since it is ultrasonic waves, they are produced in such a high energy level that it releases heat. And that heat, if suppose हमने ice नहीं रखा है, आपका ये जो tube रहता है वो भी गर्म हो जाता है, ठीक है, जिसकी वजह से the chances are that the proteins or the enzymes which are present in which you're interested may get denatured. So that's why we keep on ice. Okay. So during this sonication, a process called cavitation occurs in which tiny bubbles are formed and explode, producing a shock wave, which is going to disrupt the cell walls by the pressure change. Okay. So this is done mainly for your fungal plants में भी कर सकते हैं, हम bacterial cell पे भी कर सकते हैं. So when I was working on this, like when I have used sonicator I have used in the case of the bacterial cells. मैंने plants के case में use नहीं किया है. Plant के case में motor pestle was very easy actually to be used. So this is how the machine looks like. So here you can give the pulses. तो जैसे कि हम कैसे करते हैं, हम 10 सेकंड के लिए सोनिकेशन देते हैं, 10 सेकंड के लिए रेस्ट देते हैं, देन अगेन 10 सेकंड के लिए हम सोनिकेट करते हैं, 10 सेकंड के लिए रेस्ट देते हैं. ओके, तो इट वेरीज डिपेंडिंग ऑन योर लैब कल्चर. कुछ लोग 5 सेकंड का रेस्ट देते हैं, 5 सेकंड का ये देते हैं. सो डिपेंड्स, आप 10 सेकंड का भी सोनिकेशन दे सकते हो, 5 सेकंड रेस्ट दे सकते हो. सो इट इज टोटली ऑन यू हाउ यू वांट. ओके, so this is a beaker, ice रहेगा, suspension रहेगा, ये आपका probe आता है which is going to generate. So this is just a video where you can see how this lady or the person is doing. Okay, can you see it? These are the appendops in which the cell suspension is kept. There is no volume to this. Okay, so the shield is there. You also get ear cuffs so that you don't get into your ears and it is not damaging you. So ice here. And this is the probe you have inserted into this. Now, after this is being done, you take into a centrifuge vial. Okay, centrifuge vial we say. Then centrifuge kiya jayega. So whatever the debris are there, it will get settled at the bottom, and on the top, supernatant, you're going to get your protein suspension or the protein solution. This is the rotor into which you're going to keep your centrifuge tubes. And this is the machine. So this is your ultracentrifuge actually. Okay, clear? Sonication is clear? Okay, now coming to the homogenization. Now, till now we were talking about the bacterial cell and the animal cell. Sorry, plant cell. Homogenization is best for your animal cells. Okay. Now, why homogenization is best for animal cells? Because animal cells doesn't have that. Yes. Because animal cells don't have. Correct. Cell wall. They don't have cell wall. So it is easy. So it is easy for us, for the researchers, to carry out homogenization in the case of the animal cells. आपको छोटा सा बस उसको pressure देना है and it is going to give you the cell membrane will rupture. Okay. Okay, through a narrow space. So this is also we have done mainly for the eukaryotic or the mammalian cells. So ऐसे छोटा पतला सा रहेगा, and then you can put your sample over here, and something which is like this, a narrow tube like kind of things will be there. So slowly if you just give this, because of the shearing forces, the animal cells जो भी यहाँ पे रहेंगे, they also get ruptured. Okay. Okay. Now coming to the non-mechanical methods. So those are freeze thaw, thermolysis, osmotic shock, electric discharges, and then we have got the chemical and enzymatic methods. Now, out of this also, okay, out of this also, freeze thaw is something which you can do. Freeze thaw matlab kya hua? Directly you're putting at a very low temperature and bringing it to room temperature. उसके बाद अगेन आप उसको लो टेम्परेचर में ले जा रहे हो, उसके बाद रूम टेम्परेचर में ला रहे हो, तो जिसकी वजह से क्या होगा, द सेल मेम्ब्रेन और सेल वॉल बिकम्स लूज, ओके, एंड later on you can use the mortar pestle in order to rupture the complete cell. Okay, so this I have done when I was working on the bacterial predation. So we have done this thing in order to make the cell wall loose of the bacteria. I have done the freeze thaw. यानी मैं क्या करती थी, -80 में रखती थी, okay, for like 2 hours, and then suddenly at room temperature, यानी कि 37 पे, not 37, sorry, 25 degree पे, okay. So this is what we have done. So you can do like -80 पे जाओ, सीधा 37 पे भी लाओ, then again go to -80, then again bring it to 37. So this makes the cell wall loose. Okay, this is one method. I haven't used microwave or thermolysis because see, most of the research is based on the protein and DNA. So if I use thermolysis, it is going to denature my product, right? So I haven't used, and I never saw anyone using the thermolysis. Osmotic shock, yes, this also you can do if you are working on, you know, cell membrane based studies, for example, like RBC based you're studying. So you can change the osmotic solutions, like normal hypertonic to hypotonic, you can go, and this will result in the cell membrane burst. Okay, so even that you can do. Electric discharges also I haven't used, but yes, of course, you can give the electric impulses. Again, it is going to break the cell membrane, cell wall. Okay. Okay, now let's talk about the chemical and the enzymatic methods. So all this we have discussed. Yeah, so chemical methods. So again, this chemical methods, we use it mainly for the plants, and it is really important. Why? Because when you start working on plants along with the proteins and DNA, plant source has got high concentration of carbohydrates. They also have secondary metabolites, correct? As compared to bacteria and animals, the amount of secondary metabolites is very high. So इससे क्या होगा? It is going to affect the protein purity or DNA purity. Now, why the question is coming is because, see, you know that at 260 nanometer, it is all your heterocyclic bases which absorbs the, which gives the absorbance, correct? Are you with me? Able to follow?
Yes, ma'am.
Now the secondary metabolites are also made up of phenols. They're also made up of cyclic compounds. So they will also give readings at 260 nanometer. Okay. So if suppose you're doing the isolation of DNA from plant material, and you see that at 260 nanometer, the concentration of DNA is coming 1 gram. So don't just believe that it is the concentration of pure DNA. There might be chances that the phenolic compounds from the secondary metabolites of the plant are also present. And that is what is known as the contamination. Okay. Getting the idea over here? Like isolation is just bringing the DNA out of the cell. Purification is just having only the DNA and no any other contaminants. Okay. So there are different chemicals which we use. So there are like different organic solvents are there, phenyl, ethanol, alcohol, DMSO is also there. ADTA is also used to disrupt the cell walls of gram-negative bacteria, etc. Okay, so these are the important chemicals which we use. Now, when we talk about the DNA, RNA purification, there are many other compounds are there, phenol, chloroform, phenol, ethanol, everything will be used. Okay. Coming to the enzymatic method, this is important. So if you're dealing with plant, again, enzymes like beta, 1613, glycanases, proteases, and mannase should be used in order to disrupt the cell wall. Okay. Then you get the protoplasm and you can start working. Now, in the case of bacteria also, sometimes if you want a complete cell wall disruption, we also add lysozyme. Okay, so you know that lysozyme can break the cell wall of bacteria. Okay. Now detergent is your choice. Like if you're working with DNA, it's fine. But if you're working with protein, I suggest not to use SGS. Why? Because it's a detergent. SGS is a detergent, and detergent can denature your protein. So it is your understanding of the concepts, like when to use what. Are you getting the point over here? It depends totally whether you're working on proteins or working on DNA or RNA. Okay, depending on the requirement, you're going to add these. So अगर मैं lysozyme add करके motor pestle कर रही हूँ, so the chances of having complete bacterial cell lysis is high if only I use motor pestle method. Okay, so what I'll be doing, it's better that I add few amount of lysozyme enzyme. Agree? If I'm working on protein, then I will not add SGS because SGS will denature my protein. So I will not add SGS. Clear, guys? Able to follow me? Okay. Now, so we know EDTA, it can eliminate, it's actually your what? Cation chelator, right? So it can destabilize the outer membrane of the bacteria. So we know the calcium is required over there. So it can chelate the divalent cations from there and then start helping in cell lysis. Okay. Yes, of course. You see, if suppose I am working on membrane lipids, okay, membrane lipids, if you are not using detergents, then it will just precipitate out and it will become insoluble protein, okay, and it will be of no use. Okay, they will just aggregate and it will result in the unfolded protein. Okay, sorry, it will just go for the misfolding and the protein is of no use. But if I want it to be in proper structure, retaining the structure and activity, then this membrane lipid should be isolated with the help of the detergents. Now here we don't use SGS. You might have heard of Triton X100 or X10, something like that. Yes? Suna hai kya kabhi? हाँ. So such kind of detergents are used if suppose you're working on membrane lipids. See, previously I said if I'm working on the proteins, I won't be using. But if it is membrane lipids or membrane proteins, then I need detergents in order to isolate and purify them. Okay, it is all depending on your call, like what kind of question is there. Now working with the plant, of course, we need cellulases, chitinases in order to have the cell wall broken. Okay, C-tab is also used in the case of the plant, but it's not always required. Now, after breaking up the cell wall, the plasma membrane, now we have got the mixture. This mixture is containing of everything, DNA, RNA, proteins, lipids, carbohydrates. Okay, now depending on the requirement, we are going to have the next step. Okay. Now, suppose you're working on the DNA. Suppose you're working on the DNA, then in the centrifuge, where do you expect it to come after centrifuge? See, first centrifuge is going to be very mild of 1000 RPM so that the cell debris can be removed. Okay. After this, I'm going to increase the speed of the centrifuge. Let us say that 20,000 RPM. Okay. In this, can you just tell me at which location in the centrifuge tube I'm going to find my protein and I'm going to find my DNA? Yes, anyone?
Ma'am, protein will so it will be in the bottommost part of the centrifuge tube.
Akshay, your voice is breaking. I just heard protein and bottom. So you have got the supernatant, you have got the bottom. Okay. So where does DNA is going to lie?
DNA will be in the supernatant, whereas the protein will be always as present in the pellet region.
Okay, Akshay, you have just said the opposite. It is totally opposite. Bottom, it is going to be your DNA because it is heavy. Your DNA is going to be heavier than the protein. So protein is going to be in the supernatant every time. Okay, so once the cell debris are removed and if you increase the speed of the centrifuge, DNA is going to be heavy as compared to your protein, always. In between, you can have mitochondria, chloroplasts present over here. But if the question is coming about the DNA and protein, then it is the DNA which is present in this bottom, and in the supernatant, it is the protein. So you have such a huge DNA onto which your proteins work, right? So which is going to be more denser? DNA, right? Any doubts are there on this?
Ma'am, while working with
Hello?
hello, ma'am.
Ah.
When working with the bacterial culture in order to isolate proteins from it, people usually centrifuge and use only the pellet for further purification process.
See, that's what I'm saying. When we are going to come into the DNA purification, then it is going to change because it depends on what you have added. We'll come to that. We'll come to that. Right now, we are just doing the isolation of the stuff. We are not doing the purification. Okay. When we start doing the DNA purification, when I say purification, then I don't want carbohydrates. I don't want proteins. So that time I need to throw them out. To throw them out, it should be present in the pellet. So it depends on what chemical I have added so that the protein gets precipitated out and DNA remains in the supernatant. Okay. But if the question is based on the density, okay, depending on the density, DNA is much heavier than your proteins. Okay. In the isolation step. We'll come to that. We'll come to that. Okay. So now it is clear?
Yes, ma'am.
Okay. So this is just a video to show how animal cells are being used for the disruption and to get the lysate. When you say lysate means you have just disrupted the animal cell membrane. One second, let me just see whether volume is there or not. Okay, just the music only. So right now what we have done, so in the animal cells, you know that it is present to the bottom surface, your cells are attached. Okay, it is called adherence. Okay, so cells are attached like this, and on the top, this pink color thing is the media. So now this person is just removing the media. TBase is your phosphate buffer cell line. It is required to wash so that if any remaining media is present, that can be removed. See, now what they have done by pipetting more and more. Okay, so either you can do like this. This is also it's a part of mechanical, and you can also add enzyme treatment. Also, you can give. So in the case of animal cells, you can give trypsin. So what does trypsin do? In the case of animal cells, the attachment of the animal cell to the bottom of this surface is via the help of the proteins. So that protein attachment you can remove with the help of the trypsin. This step is also known as trypsinization. So this will help in removing the attachment of the animal cells onto the surface. So what will happen now? The cells are present in the suspension. Okay. Now it is put into the centrifuge tube, into the centrifuge. Okay, can you see it? It is turbid in nature. Supernatant is removed, and then whatever is required, it is present in the bottom. So you have taken the cells. The cells were present in the bottom, and rest of the supernatant you have removed. Now what you have done, you have added the lysis buffer. Okay, so in the case of the bacterial cell and plant cell also, you can have the lysis buffer into which where we add the lysozyme in the case of the bacteria. In the case of the plants, you can add cellulases, you can add pectinases in the lysis buffer. And in the case of the animal, they have shared this thing. See, you can see buffer is there, NaCl is there, EDT is there. Then we have got the Triton X under the detergent. SGS, can you see the percentage? It's very, very mild. Okay. And then the protease inhibitor. Why protease inhibitor? Otherwise, the proteases which are present in the cell cytoplasm is going to degrade the other proteins. And if suppose I'm working on the proteins, then it will be of no use if the proteases of the cell has degraded the other proteins. Okay. Now incubate it for some time. Vortexing. Now you can put it for ultracentrifuge. So at the end, after the centrifuge, at the bottom you're going to get the cell bubbles, and on the top supernating you're going to have your lysate. Okay, guys, clear? Is it clear?
Yes, ma'am.
Okay.
Yes, ma'am.